Hey everyone. So in this video, I'm gonna first change my color because I just like to have that darker color. I really don't like to use red as much. In this video, we're going to talk about the structure of DNA and DNA replication. So we talked about how the structure of DNA was determined in our last video. So in this one, we're going to look at what DNA looks like actually. So it is a chain of nucleotides. The nucleotides or a nucleotide contains a phosphate group, a deoxyribose, that's that pentose sugar, and a nitrogenous containing base. The bases are adenine, guanine, thymine, and cytosine. And adenine always pairs with thymine. Guanine always pairs with cytosine. Remember, that's Chargaff's rule that we learned in the last video as well. It is a double-stranded chain, and it is formed in a helical shape. And we know that it's helical because of the X-ray diffraction picture that Rosalind Franklin did. So a lot of people helped in determining that structure of DNA. Oh, hello. Let's go. There we go. So if we unwind the DNA, it looks like a ladder. And so when you have the DNA wound up, think of a ladder that is kind of wound up, or a staircase that is helical, that winds up going upwards, right? The sides of the ladder are the sugar and phosphate backbone. So you have phosphate, sugar, phosphate, sugar, right? And these, so this group here and this group here would be bound together via a bond known as a phosphodiester bond. So the rungs of the ladder are bonded through a covalent bond. Not the rungs, I'm sorry, the side of the ladder, not the rungs. The side of the ladder are covalently bonded, and the bond is called a phosphodiester bond. Hold on one moment. The two strands of the DNA are anti-parallel, which means that they're going to be running in the opposite directions of each other. So think of one strand running upwards and the other one running downwards, okay? The sugars on each strand are then going to be oriented oppositely. There's a 5-carbon atom is the uppermost one on the strand, and the 3-carbon atom is the uppermost on the other strand. So, and let me show you what that looks like in this other picture. So here you see that 5-carbon end right there, and here is that 3-carbon end. So they run in opposite directions. One runs right here, you have the 5' to 3', and this one runs 5' down here to 3' up here. So they're anti-parallel. Also notice that when thymine and adenine bind together, there are two bonds that hold them together. These bonds are called hydrogen bonds. Remember, hydrogen bonds are very weak bonds, but when you have multiple hydrogen bonds bonding things together, that's going to become very strong. So when you have hundreds and hundreds of hydrogen bonds holding your DNA strands together, it can hold them together very effectively. For adenine and thymine, there's always 2 hydrogen bonds, and in guanine and cytosine, there are always 3 hydrogen bonds. DNA then has to replicate before the cell can replicate. So every single cell has DNA in it. And for that to occur, anytime we produce new cells, we have to first replicate our DNA. So we have a copy of every single DNA molecule in that new cell. Replication of DNA then is called semi-conservative. And the reason for this is when you have DNA, you have your parent strands here, one running 5' up here and 3' down here, the other running 3' up here and the 5' down here. So when they are replicated, they produce daughter strands. And so I'm going to draw this one over here, okay, and then this one's going to go over here. And the daughter strands, I'm going to make in light blue, are going to be complementary. So this daughter strand would have a 5' end here and a 3' end there. This daughter strand over here would have a 5' end here and a 3' end there. And then the parent and daughter strand wind up together. So that is why it's called semi-conservative, because you have an old and a new strand together. So what happens in replication? The first thing that has to happen, all right, before replication begins, the DNA strands are hydrogen bonded together and they're wound up. Whoa, that was wild, huh? So the first thing that happens is DNA is going to get unwound and then it's going to get unzipped. And that should actually be Z-I-P-P-E-D. I just realized there's a P missing. Sorry about that. And then new complementary DNA nucleotides are going to come in and they're going to bind to their proper space. So if there's a T in the parent, then the daughter strand will get an A. If there's a G in the parent, then the daughter strand will get a C. So that's going to allow for a proper base pairing. Then any breaks in the sugar phosphate backbone are going to be sealed up with an enzyme called DNA ligase. And then the double helix will wind back up. And you'll have two molecules that are identical to each other, each consisting of one daughter and one parent DNA strand. Oh, sorry about that. My whole computer just went up and down. All right, so here we have the parent strand is in blue here and here, and then the daughter strand is in green down below. So you see DNA unwinding and unzipping. Unzipping means the hydrogen bonds are broke. And then we have these nucleotides coming in. What allows for the DNA nucleotides to come in? An enzyme called DNA polymerase actually binds to the DNA molecule on each side. They bind to the 3' end, and then they produce new daughter strand DNA, allowing new nucleotides to come in and bind. And so what you end up having is this identical copy of DNA consisting of one parent and one daughter strand. So remember, the enzyme that did this is DNA polymerase. So here you can see DNA polymerase binding. DNA polymerase binds to the 3' end of the DNA only. So let me write that up here. DNA polymerase binds to 3' end. And we only produce DNA then in a 5' to 3' direction. And so one strand of DNA is going to be synthesized continuously, and that's called the leading strand. So here you can see the continuously synthesized strand. And then the other side is going to be produced discontinuously. So this discontinuous replication occurs on the lagging strand. And the reason for discontinuous Replication is because when you open the DNA, remember, one side is going to have a 5' here and a 3' down here. The other will have a 5' down here and a 3'. So it's wound up. If we start opening it on this side, going in that direction, you can bind DNA polymerase to the 3' end right there and produce a new DNA strand continuously. But on this side, you can't, because you can only bind to the 3' end. And so what happens is DNA polymerase will bind here and produce a small section. And then as it unwinds more, DNA polymerase binds here and you produce another section. Those small sections of DNA that they're producing are called Okazaki fragments. So named for the individual who found them. So all of these Okazaki fragments then are going to be bound together by DNA ligase. And so you'll end up with identical daughter DNA molecules, but one is continuously synthesized, the other is discontinuously synthesized. Apologies, that's our dog, Frank. He's crazy. So this is my replication. I just kind of thought this would make it more understandable. So here we have the 5' end of DNA. Down here's 3' end. They were bound together initially. So adenine and thymine has 2 hydrogen bonds. Anywhere that you have cytosine or guanine, there was 3 hydrogen bonds. I've unwound and unzipped the DNA, okay? This is DNA polymerase. So let me just draw a line and say DNA polymerase. Binds to both sides on the 3' end, okay? Now, what's happening is DNA polymerase allows new nucleotides that are complementary to the parent strand to bind. So anywhere we have adenine, thymine comes in. Anywhere we have thymine, adenine comes in. Anywhere we have guanine, cytosine comes in. Anywhere we have cytosine, guanine comes in. And this is going to occur all the way until we have completely replicated the DNA strand. This here is going to be the 5' end. This is going to be the 3' end. This here will be the 5' end. This here will be the 3' end. And so that is DNA replication. I'm actually going to stop here and we'll get into gene expression in our next video. So have a great day. Bye.